Good. Thanks, Justin, for a very well researched introduction. So today I'll be talking to you about cause expression and that's mentioned. This is really a new concept you've developed in the past 2 years working with Desi nodes. So before I introduce cause expression as a concept, let me just introduce a little bit about spatial path economics and what the fundamental problem is. So each cycle that I get you are essentially gene counts in individual cells, as well as the spatial population gets it do. And what the fundamental question that really comes to mind is, okay, how do cells coordinate their gene expression? So for example, how is gene expression, orchestrated between nearby cells? And the current approaches are tend to be, let's select a couple of ligand receptors we already know about, cell types that tend to be expressed in, and see if their occurrence, they're essentially co-localized within a spatial domain. But the way we want to really do this is to say, in your gene panel, how many pairs of genes there are, and every single cell resolution, whether those pairs can be coordinated again and again and again. So that's that's the simplest model, but no simpler than that. So that's where you want to go with cross expression. So co-expression, as I'm sure you're familiar, just refers to the expression of two genes in the same cell again and again and again. Cross expression you define as mutually exclusive expression of two genes across spatially neighboring cells. So that's a kind of advantage of spatial path economics, where you have information about which cells are physical neighbors in a tissue space. So the way we are trying to make the algorithm is to say, okay, suppose you have two genes, the red one and the blue one. Some cells express the red gene, other cells express the blue gene. You sample cells that are red, and you ask if their neighbors tend to express blue cells. And you can calculate the probability of that given the incidence of those cells in the population at large, of those genes in the population at large. And we model it with a heterogeneous distribution, and that gets you a p-value per gene pair. So in however many genes you have in your gene panel, you can then answer the question, amongst all the pairs, which one tend to show their statistically significant spatial coordination across the gene? Okay, so as an example of what we found, we commissioned very well-known biology, particularly from neuroscience. So where you have seen on the left-hand side, the cells are colored by the expression of the specific gene, BID1, which is the receptor for B1 dopamine and interferon, and N, which is essentially a marker for the D2 neuronal types. And you can see that, okay, you see on the left side, you can tell, okay, they're really like concentrated in the striatum. But on the right side, if you just plot those cells where those genes are expressed, you can tell they're really like enabling each other again and again and again. And it's localized within the striatum region. So it's like a bit of a sanity check there. Okay, things we do expect to see, specifically the coordination between the two dopaminergic receptors does in fact get recovered by our algorithm. So that was just one gene example. You can look through this, many you can find, but we then want to do a slightly more systematic analysis where you can make, rather than having a gene and another gene and its cross expression, you can develop a network formalism where you have nodes representing genes and edges representing cross expression between them. And in this example, we made a network from one of the data sets that I analyzed. And the most salient feature topologically that you can see is that this gene called GPR20 is smack in the middle. So then we asked, okay, what does this look like if we go back to the tissue space? And once you grab, so the first thing we did is to say, okay, let's look at all the genes. So there are many genes that are cross-expressed in GPR20. What kind of cell types they tend to be expressed in? So on the top right corner of the slide, you can see that I'm plotting the co-expression of genes that are cross-expressed with GPR20. So in other words, it's a way of saying the cells that are neighboring GPR20 cells, the nearest neighbors, whether what cell type they really are. That would be like a more sort of indirect way of saying that. And you can see that they tend to be expressed in endothelial cell types as opposed to any other cell type, meaning that GPR20 seems to be nearby what you might describe as, say, blood vessels or capillaries. And on the left side, once you plot cells that are expressing the GPR20, you see these linear streaks. So the next question was, okay, visually it looks like these are linear streaks, but if you want to get a statistic, a number one, it's saying that yes, that association is in fact significant in some meaningful way. Once you do that, on the right-hand side, you can see that once you have GPR20 cell within the neighboring vicinity, it it tends to be the case that other GPU20s are localized nearby, as opposed to if you choose a random cell. So you can call it AURC style color, RC style curve, where basically on the x-axis you got neighborland, the y-axis is a cumulative picking on the curve in the middle, and it gives you some sense of the effect side, where you can say that, okay, they do tend to be within a spatial neighborhood, and visually that spatial neighborhood looks like linear streets. So it's essentially getting to the idea that the gene that GDPR20 is cross-expressed with tend to be endothelial cells, and it then makes sense that, okay, if that's what you're cross-expressing with, you would make linear streets as GPR20 does, because that's the structure of the blood vessel, which we already know about. Next, so you can have cross-expression networks. One thing you can do afterwards is to compare different networks. So on the top left graph, what we've done is to take a mouse spring with many, many different slices, compute a cross-expression network within a slice, and then compare them across the slices and see how similar they are. And because spatial gene expression varies as a function of space, you expect cross-expression to vary as a function of depth as well. So once you thought that, similarity between adjacent networks is much higher, and as you compare more and more distant network, it goes down. So you see a nice downward sloping curve over there. In the bottom, we did another experiment where essentially we registered our data to the Allen brain atlas, which gives you a brain region anatomical annotation distribution of the hippocampus, something else is the striatum. So you take a region, you compare that region with other regions in the same brain, as well as the homologous region in a different sample, and you plot those distributions as to how similar those anatomical region-specific networks are. And you see that the homologous regions tend to be much more similar than the non-homologous region, either within the brain or between the brain. So there is some sampling variability between different brains. Nevertheless, the same regions are a lot more similar than you would expect by comparing different regions, in various ways. In the top right, this was another kind of a global analysis where we tried to look at cross-expression networks between different replicates. So in this setup, that we had 3 replicates, and we compared it, and you can see it's pretty strong correlation between their cross-expression networks. And finally, we developed a software where all you have to do is put your gene expression matrix and your cell coordinates, metadata, and the output would be a gene by gene matrix where each value is basically a p-value, which tells you whether or not those 2 genes are associated across the tissue. And this runs pretty fast. So I did a validation assessment on my own laptop, just a regular 16-GB laptop. And if you have, say, 5,000 genes, then you're like, I have a slice with 200,000 cells. It runs in under 10 minutes. So it runs pretty fast. So if you put it on a computer, on a kind of a cluster, the data sets get bigger and bigger and bigger. This would scale very, very easily. So as I was talking with Justin about it, basically our preprint also came out in the meantime. So I encourage you to take a look at it. If you go to our archive and type cross-expression, it's the first hit, so you'll find it easily. And I also had we've developed a GitHub page for a manual which walks you through how you can implement some of these analysis. In addition to just the very fundamental cross-expression p-value analysis, there's a whole bunch of ways you can visualize the data. The thing that showed you about 2 genes being next to each other, you can put any gene here, and it will make a plot where you can show cross-expression, you can show their expression, where both genes are expressed, and so on. So there's a lot of ways you can analyze your data using some of the tools that we've developed. And they get appeared, obviously, in the preprint made as well. And finally, I'd like to acknowledge Jesse for supervising my project and developing the framework for the past 2 years, Charlie Chen and his lab, Mara and Helmi, for collecting and giving us the bars data that we use for allowing the analysis in this paper, and Leon for discussing all things data analysis and spatial possibilities with me. And finally, some funding acknowledgements, a fellowship from UP and answer grant, allowed us to do this stuff. And Jesse, Shavina, and Leon would like to acknowledge NIH funding from the listed grants as well. Thank you.